Hi everyone, hi sir. We are the group 11 reporter about translation in Eukaryotes and Prokaryotes. But before that, I am Willie Indilatori and I am the first reporter. So again, this is our topic, translation in Eukaryotes and Prokaryotes. So what is Eukaryotes and Prokaryotes? Eukaryotes are organisms with a complex cell or cells, in which the genetic material is organized into a membrane-bound nucleus or nuclei, and it also contains organelles. So it means eukaryotes from the word complex itself, meaning it is not easy to read or to process. Then, prokaryotes. It is any organism that lacks a distinct nucleus and other organelles due to the absence of internal membranes. So, bacteria are among the best-known prokaryotic organisms. So, prokaryotes, it is lacking with nucleus and it composes only few of organelles. So, this is the illustration between prokaryotes and eukaryotes. In a simple explanation about the illustration, prokaryotic cells are cells without nucleus, and eukaryotic cells are cells that contain nucleus. Eukaryotic cells are cells have other organelles beside nucleus. The only organelles in a prokaryotic cell are ribosome. So, what is translation? Translation is basically a synonym process of protein synthesis. I know that you are already aware about protein synthesis. It is the process in which the protein is synthesized from the information contained in a molecule of messenger RNA, meaning from DNA, if it will unzip, it will transcribe into mRNA, and after transcription, it will translate into protein. Then, it can be defined as the process by which the sequence of nucleotides in a messenger RNA molecule directs the incorporation of amino acid into protein. So based on the picture, nag-start siya sa DNA with 2 strands. Then, pag undergo niya sa transcription process, the RNA polymerase will unzip the 2 strands or it will become separated. After that, the mRNA will enter the ribosome producing protein. So let's proceed to the translation machinery. The machinery required for translating the language of messenger RNAs into the language of protein that is composed of 4 primary components. So these are the 4 primary components of translation machinery. The first is the mRNAs, second tRNAs, third enzymes, and then the last is the ribosome. So mRNAs or messenger RNA provides an intermediate that carries the copy of a DNA sequence that represents protein. So ang messenger RNA sadya ang molecule na nagdaya ng genetic information na kinahanglanon para sa pag-produce ng protein. Next, the tRNAs. It acts as an adapter between the codons and the amino acids they specify. So, ang transfer RNA sadya an adapter between the genetic instruction written in the nucleic acid sequences and the protein product encoded in the genes. And next, the enzymes. It required for the attachments of amino acids to the correct tRNA molecule. So, meaning, sadya ang kinahanglanon para sa pag-attach ng amino acids to the correct tRNA molecule. So, there are 2 parts of enzymes, the amino-acyl tRNA synthetase and then the peptidyl transferase. So let's proceed first to the amino-acyl tRNA synthetase. It is essential and universally distributed family of enzymes that plays a critical role in protein synthesis, pairing tRNAs with their cognate amino acids for decoding mRNAs according to the genetic code. So meaning, sadya an essential na molecule for the pairing of tRNAs sa ila kaugnay or kaparihan na amino acids para mabasa ang mRNA according sa ila genetic code. Then, the peptidyl transferase. So, ang peptidyl transferase sadya ang responsible for peptide bond formation during the protein synthesis. And the last part of the translation machinery, ribosome, it is the macromolecular complex that directs the synthesis of proteins. So dito mo singod ang genes para makaproduce ng proteins. Again, based on the picture, mRNA, as what I have said that sidya an molecule na nagdaya ng genetic information na kinahanglanon para sa pag-produce ng protein. And then, ang ribosomal RNA, sidya an nag-ensure na sakto ang alignment ng mRNA and ribosome during protein synthesis. And then, the transfer RNA or tRNA, sidya an adapter between the genetics instruction written in the nucleic acid sequences and the protein product encoded in the genes. And now, let's move on to the translation process. In prokaryotic cell, transcription and translation are coupled. So that is, translation begins while the mRNA is still being synthesized. In a eukaryotic cell, transcription occurs in the nucleus and translation occurs in the cytoplasm. So, ang transcription, pag-abot sa eukaryotic cell, ato siya musingod sa nucleus. However, ang translation, ang eukaryotic cell ato musingod sa cytoplasm. So again, in the translation process, it involves the three major steps: the initiation, elongation, and termination. Let's move on to the initiation. The initiation of translation, if eukaryotes is complex, involving at least 10 eukaryotic initiation factors or ELFs and divided into 4 steps. The first step is ribosomal dissociation. The second step is formation of 43 subunits pre-initiation complex. Then, the third step is formation of 48 subunit initiation complex. And the last is the formation of 80 subunit initiation complex. So, let's come now to the first step, the ribosomal dissociation. So, as you can see in the picture, there is 80 subunit ribosome, the 60 subunit, and then the 40 small subunit, the initiation factors 1A, and then the 3. So, the 80s ribosome dissociates to form 40S and 60 subunits. So, an 80 small subunit, pag once ma-dissociate siya, maka-form siya ng 60 subunit and 40 small subunit. So, the two initiating factors, namely ELF3 and ELF1A, bind to the newly formed 40 subunit and thereby block its reassociation with 60 small subunit. So, in eukaryotic initiation factors 1A and 3, pag once na mo-bind siya diri sa 40 small subunit, siyaan mag-block nan inin 60 subunit para dili sa maka-reassociate sa 40 small subunit. So let's come now to the next step, the formation of 43 subunit pre-initiation complex. A ternary complex containing methionine, tRNA, and eukaryotic initiation factor 2 bound to GTP attaches to 40 subunit ribosomal subunit to form 43 subunit pre-initiation complex. So from methionine and initiation factor na 2, mo bound siya sa GTP. And then after ining methionine sa initiation factor na 2, mo bound sa GTP, ang GTP mo attach siya sa 40 subunit. Then after niya mo attach sa 40 subunit, maka-form siya ng 43 subunit pre-initiation complex. So the presence of eukaryotic initiation factor 1A and eukaryotic initiation factor 3, sila ang nag-stabilize ng dyaon na complex. So meaning, sila ang nagpasulid sa pag-process ng dyaon na complex. So let's come now to the third step, the formation of 48 subunit initiation complex. The binding of mRNA to 43 subunit pre-initiation complex results in the formation of 48 subunit initiation complex through the intermediate 43 subunit initiation complex. So from mRNA methionine to 43 pre-initiation complex, pag once mag-bind sila, maka-form ng 48 subunit initiation complex. So, eukaryotic initiation factor 4F complex is formed by the association of eukaryotic initiation factor 4G eukaryotic initiation factor 4A with eukaryotic initiation factor 4A. The eukaryotic initiation factor 4F, or referred to as cap binding protein, binds to the cap of mRNA. Then, eukaryotic initiation factor 4A and eukaryotic initiation factor 4B bind to the mRNA and reduce its structure. This mRNA then transferred to 43 subunit complex. So, for the appropriate association of 43 subunit per initiation complex with mRNA, energy has to be supplied by ATP. So, from gikan sa 43 subunit per initiation complex with mRNA, ini 43 subunits pre-initiation complex with mRNA methionine, ang nag-supply sa ilang ng energy is the ATP. The ribosomal initiation complex scan the mRNA for the identification of appropriate initiation codon. So, ang ribosomal initiation complex, siya ang nag-scan sa mRNA methionine para sa pag-identify ng appropriate initiation codon, for example, like EUG. 5' EUG is the initiation codon. So therefore, the 5' EUG is the initiation codon. So let's proceed to the last step, the formation of 80 subunit initiation complex. 48 subunit initiation complex bind to 60 subunit ribosomal subunit to form 80 subunit initiation complex. So, from 48 subunit complex initiation complex, mo-bind siya sa 60 subunits. Pag makabind na siya, maka-form siya ng 80 initiation complex. Then, the binding involves the hydrolysis of GTP bound to eukaryotic initiation factor 2. This step is facilitated by the involvement of eukaryotic initiation factor 5. As the 80 subunit complex is formed, the initiation factors bound to 48 subunit initiation complex are released and recycled. So, pag once an 80 subunit complex ma-formed, the initiation factors bound to 48 subunit and complex ma-release tapos ma-recycled.
As the continuation of our report, I am Ms. Mary Rose Elandang Makabudbud, and I'm going to discuss the next step of translation in eukaryotes, which is the elongation and termination, and also the ribosomal recycle. Let me first emphasize that in eukaryotes, there are 60S subunit, and in eukaryotes also, there are two elongation factor called elongation factor 1A and elongation factor 2. Ribosomes also have three important binding sites, the E site, P site, and E site. The E site or acceptor site is used for the attachment of incoming amino acid transfer RNA. Now, the second site is the P site called peptidyl site or parking site. This site occupied by peptidyl tRNA, which is the peptidyl tRNA carrying the growing polypeptide chain. Now, the last site is the E site or the exit site, and this is the site where the empty tRNA occupies prior to exiting the ribosome, and it is also called deacylated tRNA because it does not have any amino acids. So, when transfer RNA carrying amino acid, it called amino acyl tRNA, but if it does not contain amino acid, it is called deacylated tRNA. So we end up in the initiation in ATS ribosome complex, where the AUG is the start codon and it carries methionine, so we have the P site occupied by the transfer RNA, and E site and E site are both empty. The 60S ribosome subunit contain a factor binding region, and this complex moves on the next step where the amino-acylated tRNA is carried elongation factor 1A, which just carried GTP. GTP means guanosine triphosphate, and it is hydrolyzed and it releases energy, and that factor utilized for elongation factor assembly. Now, this ternary complex, the elongation factor 1, binds near the 3' of the transfer RNA close to the amino acid. Now, the complex that results where elongation factor 1A is positioned near the factor binding region, and the E site is still not occupied by anything. One detail worth mentioning in this step is that the codon is partially controlled by the 18S ribosome RNA, which is part of our 40S ribosome subunit. Specifically, the 18S ribosome RNA stabilizes the codon and anticodon pairing. So when codon and anticodon stabilize the SRL region in the factor binding region, diriya na part sa atong factor binding region, it will activate the GTP, and then phosphate is released gikan sa atong elongation factor 1A. This causes the elongation factor 1A to be in the complex of GDP, and as a result, the elongation factor 1A is released. In the case of elongation factor 1A, the conversion of GTP and GDP actually causes a structural rotation in the elongation factor 1A complex. So it is the rotation releases the elongation factor 1A out of the complex. This release of elongation factor 1A brings the amino acid on the E site tRNA close with the amino acid on the P site. And now the next step, as you notice that the amino acid on the tRNA is on the P site and E site is very close to each other, and E site is still empty. Alright, we also know that from the translation, the 60S subunit contains 28 ribosomal RNA, and this 28S ribosomal RNA is responsible for peptide bond formation. Following the peptide bond formation is that the methionine gikan sa P site tRNA is transferred on the tRNA on the E site is still empty. As essentially, we all have that as form of peptide bond between the amino acids. And now this complex is ready for the next step called translocation. And this step of translocation needs the second elongation factor 2. The elongation factor 2 contains a post-translational modification of one end of protein, which is known as deptamide, which is the chemical modification of the histidine amino acid of the protein. And this deptamide modification is often time required for breaking the 18S ribosomal RNA interaction with mRNA in the tRNA at the E site. So, in the 18S ribosome, in elongation factor 2 position itself at the factor binding region where the deptamide modification is close to the codon and anticodon pairing region. And it happened that E site has the amino acid chain once naturally move to the P site because there is a protein exit channel which is very close to the P site. So this causes the tRNA to move the 40S ribosome subunit rotates, and this causes the chain to move to protein exit channel. The energy for this movement is provided by phosphate bond present in GTP of elongation factor 2. So, after translocation, the AUG start codon which was had the P sites move into E site, and the E site codon moves into P site, and as the result, the associated transfer RNAs also move with them because they're paired with the codons, and now the amino acid chain can go into the protein exit channel. So the ribosome is moved by the codon, and the E site is occupied by elongation factor 2 GTP complex. This translocated ribosome now moves to its original position, and the 40S subunit rotates back and causes the elongation factor 2 GTP, which anyways has low infinity become of its loss of phosphate from the GTP to be released, and the E site tRNA is also released. And in the next step, the P site, the only occupied site where the amino acid chain is linked to the transfer RNA, and both E and E sites are empty. This situation is actually like the starting adds complex except the amino acid chain. It is a bit longer, and this is ready for the next amino acid transfer RNA to be the elongation factor 1, and which with each cycle of translocation, the amino acid chain will continue to grow until the E site encounters the stop codon, and when a stop codon is encountered, the ribosomes undergoes the termination step. In termination step, the termination occurs by one of the three stop codon, UAA, UAG, UGA. When it binds to the mRNA leading to us right into the ribosome recycling, this process involves the recycling of the post-termination complexes. There are three release factors that we should know about to help the process of release factor 1, release factor 2, and release factor 3. The release factor 1 and release factor 2 recognizes the stop codon, but the purpose of release factor 3 is to free up release factor 1 and release factor 2 from the post-termination complex on the ribosome. When the stop codon is reached, the release factor, either release factor 1 or release factor 2, catalyzes the peptide bond and P site to the molecule of the watering the E site, and peptide is hydrolyzed and dissociates. The last step is the release of the ribosome from the mRNA, which is catalyzed by the joint activity of the EFG, a translocation factor, and ribosome recycling factor. Thus, the idea of recycling is that the ribosomal subunit are split up and available to bind to mRNA to start the whole protein synthesis process over again. Thank you.
Good morning everyone, my name is Alden Hovero, and today I will be discussing the introduction to translation in prokaryotes. Since the previous reporter discussed the translation in eukaryotes. So what is prokaryotes? Prokaryotes represent bacteria, mycoplasm, blue-green algae, and it is a Lackwell defined nucleus and the presence of cell wall. As you can see in this picture, you can see what it is in prokaryotes and eukaryotes. So there's a table 2823, the rules of bacteria and eukaryotic initiation, factors in protein synthesis. We have here the bacteria and the eukaryotes. As you can see in the table, there is a I factor, the first factor, the second factor, and the third factor, and the function. So the first factor is stimulates activities of IF-2 and IF-3. The second factor is facilitates binding of F-methionine to tRNA to 30 subunit ribosomal subunit, and the IF3 binds to 30S subunit, prevents premature association of 50A subunit. So the factor here in the eukaryotes, the function is facilitate binding of initiating methionine tRNA to 40S ribosomal subunit. The first factor to bind is a 40A subunit. Then facilitate subsequent steps. Bind to 5' cap of mRNA, bind to mRNA, and facilitate scanning of mRNA to locate first the start codon, which is the AUG. Then promotes dissociation of many other initiation factors from 40S subunit as prelude to association of 60A subunit to form 80S sub-initiation complex. Facilitates dissociation of inactive 80A subunit ribosome into 40A and 60A subunit. So later on, the next reporter we will be tackled the process of translation, the initiation, which is reporter could mention this ATA subunit and the ribosomal subunit. So the last part that I will be discussed or tackled is the genetic code. How can we read the genetic code? In this chart, we can see every possible codon nucleotides combination and the amino acid. Each codon corresponds to the amino acid. In this chart are listed using their 3-letter codons. For example, AUG. For example, MET. The AUG is the MET is the 3-letter code for methionine. In amino acid, no, this code is the same code used in translation for all living organisms. In this way, you're seeing here is universal. Genetic code is universal. When we look at this chart, we notice a few things. First, code is redundant. Multiple different codons will code for the same amino acid. You can see that the codons UUU and UUC both will code for the amino acid PH, or which is the phenylalanine. We also notice that the code is non-ambiguous. Each codon codes for one and only one amino acid. There's no question which amino acid is going to be coded for by a single specific codon is the mRNA. A few other things you may notice in this genetic code is that we have the start and stop codon that signal the beginning and the end of the coding portion of the mRNA strand as you scan mRNA molecule for translation. It will not start until you find a start codon, which is always the codon AUG for methionine. This means that every polypeptide coming out of translation will have methionine as its first amino acid. This methionine may be removed before the protein folds from the start codon. You move through the mRNA reading and translating codons one after another until you may find one of these 3 stop codons, the UAA, UAG, and the UGA. Stop codon signals the end of the coding portion of the mRNA and at the end of translation.
So hi everyone, for the continuation of our report, I am Ella Flor Navarro Polvorosa and I'm going to discuss the translation process in prokaryotes. So we all know that translation is the process whereby the base sequence of a mRNA is converted into the amino acid sequence of a polypeptide. This process involves an mRNA, tRNA, amino acids, a ribosome, and other accessory proteins. mRNA consists of 3 main parts. The portion from the 5' end to the translation start codon is the untranslated leader region, or simply called the leader. The central portion is called the reading frame. It commences with the start codon AUG, followed by a series of 3 nucleotides codons, each of which specifies an amino acid, and it ends with one of the stop codons UAA, UAG, UGA. The final portion of mRNA consists of the section from the stop codon to the 3' end called the 3' untranslated trailer sequence, more simply called the trailer. Transfer RNA functions to bring amino acids to the growing polypeptide chain during translation. It consists of a single RNA chain of about 75 to 90 nucleotides, each tRNA having a different sequence of bases. After it is transcribed, the tRNA folds up into a characteristic clover leaf structures. The anticodon contains a 3-nucleotide sequence that is complementary to a codon on the mRNA. The amino acid is attached to the 3' free end at the top of the clover leaf. The ribosome assembles amino acids into a polypeptide. Ribosome consists of two parts, the large subunit and a small subunit. Ribosomes are made of ribosomal RNA and various protein. The active ribosome contains 3 tRNA binding sites, E for exit a site, P for peptidyl site, and A for amino acyl site. In prokaryotes, translation initiation occurs in 3 steps. First, the small ribosomal subunit or 30S subunit complexes with 3 proteins or initiation factor. Initiation factor 1, initiation factor 2, and initiation factor 3. A molecule of GTP also attached. The ribosome IF-GTP complex recognizes the sequence in the leader upstream of the EUG start codon. This sequence is called the Shine-Dalgarno sequence. This sequence is complementary to a sequence at the 3' end of the 16S ribosomal RNA of the 30S subunit and serves to align the ribosome on the mRNA in its proper reading frame. Second, translation begins at the start codon AUG, a few bases downstream of the ribosome binding site. The first AUG in protein specifies a modified form of methionine. So, this modification consists of the addition of a formyl group to methionine, making it formyl methionine or FMET. The anticodon in this initiator tRNA containing FMET complements AUG. Upon binding of FMET tRNA to mRNA, IF3 is released with the remaining assembled components called the 30S and initiation complex. Lastly, the large ribosomal subunit or 50S subunit binds to the 30S initiation complex. GTP is hydrolyzed and IF1 and IF2 is released. The final complex is called the 70S initiation complex. The FMET is now situated in the P site of the ribosome with its anticodon hydrogen bonded to the mRNA start codon. Initiation is now complete and elongation follows. So once translation initiation is complete, elongation begins. The elongation phase consists of 3 steps: binding of a tRNA to the ribosome, formation of peptide bond between adjacent amino acids, and movement of the ribosome to the next codon. Elongation continues 1 codon at a time until a stop codon is reached. As initiation concluded, FMET tRNA was hydrogen bonded to AUG in the P site of the ribosome. The next codon is in the A site. In this case, the codon is UCC, which encodes serine. The serine-bearing tRNA in the cytoplasm forms a complex with elongation factor TU and GT. So this serine tRNA EF-TU-GTP complex enters the A site where the tRNA anticodon forms hydrogen bands with the UCC codon. Upon binding, the GTP is hydrolyzed and EF-TU is released and recycled. So when the tRNA in both the P and A site, a polypeptide bond can form between adjacent amino acids. This reaction is catalyzed by peptidyl transferase, residing in the large ribosome subunit. The first step involves breaking the bond between FMET and its tRNA in the P site. Next, a peptide bond forms between the now freed F-met and the serine, which is attached to its tRNA in the E site. Once the polypeptide band has formed, there remains an uncharged tRNA in the P site, and a tRNA with a growing polypeptide chain in the E site. The ribosome now moves one codon along the mRNA with the help of EFG and another GTP. An EFG-GTP complex binds to the ribosome, hydrolysis of GTP occurs, and translocation begins. The uncharged tRNA moves from the P site into the E site, blocking the next amino acyl tRNA from attaching to the A site until translocation is complete. The tRNA in the A site, still bound to its anticodon, now moves into the P site. Once this tRNA is bound correctly in the P site, the uncharged tRNA is ejected from the E site. The A site is now open for the next amino acyl tRNA to enter, allowing the elongation process to continue. Elongation continues until the ribosome reaches one of the three stop codons, UAA, UAG, UGA. Unlike other codons, stop codons do not code for any amino acid, so no normal tRNAs have anticodons that complement these codons. The ribosome recognizes polypeptide chain termination with the help of proteins called release factors or RF. RFs read the stop codon and initiate a series of specific termination events. E. coli has three RFS, RF1, RF2, and RF3. RF1 recognizes UAA and UAG, while RF2 recognizes UAA and UGA. RF3 does not recognize any stop codons but stimulates the subsequent termination events. Elongation and ribosome translocation result in a stop codon entering the vacant A site. RF1 or RF2 binds to the stop codon, which triggers peptidyl transferase to cleave the now complete polypeptide from its tRNA located in the P site of the ribosome. The tRNA is then displaced from the P site of the ribosome, followed by disengagement of the RF and 70S ribosome from the mRNA and separation of ribosome into individual 30S and 50S subunit.